So it's my pleasure to introduce Dr. Donald Tomalia. Don is the CEO and founder of Synthons LLC, National Dendrimer and Nanotechnology Center. He's also a distinguished visiting professor at Columbia University and adjunct professor at Pennsylvania. Don, talk is Precision Medicine by Design, the first Phase 3 FDA clinical approval for dendrimer-based nanomedicine guided by CNDP engineering.
Thank you, Nell, for a kind introduction. And before I start, I'd like to thank Bert Leffler for the kind invitation to share this short story with you, and also thank Anil Patrick for that nice introduction he gave earlier this afternoon, and his excitement about the first approval for a dendrimer-based nanomedicine. And hopefully the story will show you a little bit more detail about why there's all this excitement this last year. We only have 20 minutes, and so it's broken down into 4 short sections. The first being a brief historical overview of the whole dendrimer area in a matter of a few minutes. Engineering, CNDPs, it's a topic I've talked about here for the last 8 or 10 years, and I would like to share with you and show you how it was used to optimize the present dendrimer conjugates for the approval they got this past year, and show you some preclinical failures that possibly happened, lessons to be learned, because they did not optimize these critical nanoscale design parameters, or the nanoparticle or dendrimer quality was not up to standards. And then finally, I'd like to give you some real brief examples on how Star Pharma used these patents. We had nearly 200 of them between Dow Chemical. I worked for Dow for nearly 30 years. All my patents from Dow, plus other personal patents I got after leaving Dow in the 90s, were transferred to Star Pharma, and they constitute the protection for all of their products that are coming through the pipeline and or going into clinical as we speak. And I might add, AstraZeneca has a representative here that I missed, who really could tell parts of this story in more detail than I will. And that's Marianne Ashford. And anyone that heard her earlier this afternoon probably knows a lot more than I'm going to show you this afternoon about how Star Pharma is progressing in this area. Finally, some conclusions. Now, very quickly, dendrimer historical overview. It's a 30-year, rich 30-year history going back from the early 80s to the present. I'm only going to concentrate on the last 18 or 20 years of involving the certain key things that happened, such as we realized in the last year from one of our major pharmaceutical clients that their in-depth literature search showed that we were perhaps the first to ever actively target cancer tumors in animal model systems using monoclonal antibodies and dendrimers. We'll show you that shortly. We'll talk a little bit about transferring these patents to Star Pharma, and a little bit about the CNDP concept for optimizing these parameters, and a little bit about lessons to be learned on how it's necessary or it's really worthwhile optimizing CNDPs as you go to clinic. Now, this was a remarkable revelation to us this last year, and I won't name the pharmaceutical company, but they did a very thorough search of the literature, and they said, Tomalia, do you realize, as far as we can tell, first time anybody ever targeted a nanoparticle, in this case a dendrimer, in live animal models, and successfully had them localized in a tumor. Was done clear back in the 90s. We did the work in 89 to 90. It actually got published in 94, and it involved the in vivo localization of this labeled indium using a starburst antibody conjugate. The reason I show you this, it's exactly the same conjugate that Star Pharma is using today, and it's in phase 1 and phase 2, and they are working on this in collaboration with AstraZeneca. Marianne Ashford is somewhere in the audience, and so I show you this cartoon example because it's a part of the story as we go on. Now, engineering CNDPs. There may be some in this room that don't know what CNDPs are, but some of the old crowd has been listening to me talk about this for the last 8 or 10 years. And I am a very strong advocate of optimizing CNDPs. Nell knows what they are about because he hosted me at UCLA, and we talked about all these critical design parameters and how they might be used in his area as well as in the area of drug delivery. But anybody, and I know most of the people in this audience knows that the 3 key phases that one has to get through for FDA approval and clinical approval involves a phase 1, which is fairly easy, just have good safety margins, good nanotoxicity. But the phase 2 is really a tough one because you really have to show you got something that is cost effective and better than anything that's out there. And that's where CNDPs really show their value, because if you do them right, you will get the most optimized candidate to be very successful for that phase 2. And of course, phase 3 is just taking those candidates that survive phase 2 and exposing them to a more diverse population. And phase 4, of course, is for the lawyers. They're waiting around to see which ones have flaws so that they can sue a company for some victim. And so that's pretty much sums up everything that goes on for phase 1, 2, and 3. Now, how do we optimize for this phase 2? And it's very simple. Don't get lost by this eye chart, because it really is showing you that there are only 6 fundamental critical nanoscale design parameters to worry about: size, shape, surface chemistry, flexibility, elemental composition, and architecture. And the reason you want to have control over that, whether you are engineering it or whether you are looking for a nanoparticle that has all of the optimum features, is that those 6, I can't find the 7th one, those 6 CNDPs control and determine these 4 important pharmacodynamic properties, namely excretion modes, nanotoxicity, pharmacokinetics, and biodistribution. So if you can control and know and learn how to engineer those 6 parameters, you really are on your way to really having an optimized candidate for that important phase 2 clinical stage. Now, in the interest of time, Barinholtz has probably the record for the most number of liposome products that have made it through the FDA. So I didn't want to include it on the list. I wanted to focus in only on covalent organic polymers, namely synthetic polymers or biopolymers. And I'm going to just very quickly say that a number of biopolymers based on naturally on proteins, these are all optimized. The CNDPs are optimized by nature, right? They're the right size, they're the right this, the right that. Many of them have made it through that torturous challenge of the 3 clinical stages. However, synthetic nanoparticles, organic nanoparticles, namely dendrimers and polymeric micelles, require this very careful attention for CNDP engineering, and we'll focus very briefly on them as we go into the next few slides. So what were some of those preclinical failures or setbacks or momentary pauses that we can learn some lessons from? And one of the first was, and it was one that I was involved with, and it involved work with the Baker Group, and I think Anil was with us at the time. And I left shortly before this actually got taken into preclinical, but we thought we had a slam dunk for a successful candidate, only to find out that there were some totally unimagined problems that really gave them a setback at the preclinical or the phase 1 clinical stage. And just to share what we think they are in this next cartoon, and by the way, and after that we'll talk about some minor setbacks involving polymeric micelles. But let's look first at what were these lessons to be learned from this Baker episode, where he had looked like he had developed a perfectly targeting Dendrimer, generation 5 Dendrimer. He basically used the same protocol. He literally looked at our patents that I had obtained it down for that starburst, or for that antibody Dendrimer conjugate. However, what he did was to change the targeting groups to folate moieties, because many people, as you well know, cancer overexpresses folate receptors in many important cancers, and it was a very nice medical choice and decision. So he had developed a very beautiful targeting Dendrimer. However, one simple issue that he failed to take seriously, and you had many long conversations with Jim about this, and he was a physician, a brilliant physician, but he didn't always want to listen to chemistry from someone like me, and he insisted that we could get by with amide linkers or ester linkers, and I kept saying, I think we need to have cleavable linkers, because if you can target it successfully to the tumor and it can't come off, how is this active drug going to do its job? And that was one of the serious problems that he really encountered very early, close to the phase 1 stage issue. The second problem he encountered was polydispersity. And I don't know how to emphasize the importance of monodispersity of nanoparticles, because when you have different sizes of that, as you can see here, this is Mark Banasak Hall. He was a member of the Baker Group. He just published this in 2014. The exact sample, the exact material that Baker used for that clinical trial that didn't work, and what they were surprised to find out was it wasn't monodispersed. It was only about 74.9% of the desired material. It had lower materials called trailing generations, higher materials called oligomers. And when you have that distribution, these sizes are going to biodistribute to places that you may not necessarily want them to go to. So we learned an important lesson from that, as well as Star Pharma. And Star Pharma, when they make their materials, it's a single molecule. A single peak falls right here. When we make our PAM-AM dendrimers at nanosyntons, it's a single peak right here. We have essentially none of the lower or higher materials here. Therefore, monodispersity, we learn, is very important. Now, let's look at one other quick example, and it relates to an interaction I had with Bob Langer just 2 weeks ago in Boston, and he said, Tumalia, you were right. Size really matters. And if Katioka is in the audience, he told me the same thing a few years ago, but I heard his brilliant lecture just an hour or two ago, and he's overcome a lot of the problems. So what are we talking about here? Basically, it's important to have the right size, small sizes in particular, in order to get deep penetration into the cancer tumor. And in fact, this is a cartoon that shows a very desirable number is to have it no larger than 20 nanometers, or you can get some sort of EPR effect, but you will not get deep penetration that is required if you are larger than roughly 20 nanometers. And in fact, that's what I understand Professor Katioka encountered back around 2010 or so, because in 2011 he actually published in Nature Nanotechnology the importance of small size. If you don't have that small size, you don't get the deep penetration, and all you do is tease the cancer tumor. You don't completely kill it. So it comes back with a vengeance. 2 weeks ago, I had the pleasure of lecturing back-to-back with Bob Langer at the ACS meeting in Boston. And he had I made the same prediction to him many years ago, and he didn't believe it until he ran into some trouble with bind therapies. And he found that, as most people in this audience may or may not know, polymeric micelles are very difficult, if not impossible, to make smaller than 30 nanometers. And so they become almost impossible to extravagate through this pore into the deep interstitial areas of the tumor. And in fact, Bob said, you know, I read some of your papers. I didn't quite totally believe it, but he said, I do now. And in fact, during the last year or two since bind went Chapter 11. He has had his management from buying as well as his new company. He says, I don't want to make the same mistakes again that we made. They have actually been calling me and consulting with me for the last year or two. And Bob told me 2 weeks ago, he said, size, yeah, I had no idea how important that size was for deep penetration, and we're not making the same mistake again. And so that really summed up why CNDPs are important. You need to be able to engineer those kinds of dimensions, or you aren't going to get the results that you would certainly hope to. Now, Star Pharma actually saw this back in 2014. And in 2014, in their website, they actually made the comparison. They said it was not only due to size, because the bind therapy particles were 60 to 120 nanometers, whereas the Star Pharma particles, the Dendrimer particles, are 10 to 15, but they said it was also due to architecture. They were able to carry more of the drug than the bind therapies, and the other third area was they were able to really enhance the solubility of their therapy, the Star Pharma therapy, by the dendrimeric structure, and in fact as much as 20,000-fold increase in solubility versus problems that they had with the polymeric micelle. And that had to do with surface chemistry, basically. So those are lessons to learn and not to repeat. And Bob and I had some really enjoyable conversations just 2 weeks ago about all of this. And he says, that's science. That's how you go forward. You have to learn these very critical issues. Now, let's wrap up by how did Star Pharma actually use these patents and CNDPs to move forward. And if you look at their website, this is most of this is out of their website, you will see, and in fact, many people may know, that dendrimers are not only useful as nanovectors for carrying drugs in a targeted or controlled way, they also can be used as active, topically active pharmaceutical agents in their own right. In fact, their first products that were approved by the FDA really involved the use of dendrimers in that fashion. Most of this technology is based on patents that, well, I was at Dow, and as well as after Dow, we transferred over 200 patents to Star Pharma, and that constitutes their protection out to the year 2032 or 3, I believe. That whole story was written up in this Elsevier publication, Chemistry and Biology, in 2005. And I refer you to that for more details. How did they use this? They used the CNDPs by looking at sizes and surface chemistries to see how much penetration one got to the skin, because they did not want it to get into the bloodstream. They wanted it only to be active on the surface. And that was the key recipe, the key driving force behind the success of that product, and it involved having a balance between hydrophobicity and size in order to have developed and progressed VivaGel to the approved state that it's in. And as you'll see on this next slide, it's a huge market. It has a global market of $1.75 billion per year. It's the first non-antibiotic therapy to market, and it literally involved the use of many of our CND principles that I have shared with Star Pharma in the past. And by the way, in the interest of open disclosure, I own a few shares of Star Pharma, so you have to take what I'm not selling Star Pharma. I'm telling you these are the things that we shared in the past that I know led to many of their decisions on what they ended up with final structures, and this was one of the final structures they ended up. This was an active dendrimer as an active pharmaceutical. Now, 1 minute, and I'm just about done. And so as the active, they use these as active drugs in these two systems right here, either in a topical bacterial system, bacterial vaginosis system, or in the condoms. And quickly for the injectables, they looked at these CNDP relationships here in order to define their parameters for their injectables that were talked about by AstraZeneca earlier today. That story is in the website shown here. These are their pipeline of new products that are coming down based on these CNDPs and that patent position and the lesson learned, and I'm going to wrap up on time now, I hope, here. Before I give you the final lessons learned, I'd like to say a premature commentary on the future of Dendrimers in nanomedicine was made by my good friend Zonka Swinson just a couple of years ago. Little did he realize that in this last year, they finally were approved, and I had this wonderful conversation with Baron Holtz, and Baron Holtz says, Tamalia, that's just the average time it takes to get something from discovery through the clinic. He says liposomes took 34 years, and if you look at the timeline for Dendrimers, it was 32. Lessons learned, try to go for low polydispersity nanoparticles. CNDP engineering is a critical strategy that really will help you to be successful in the clinic. Use it as you're developing your standards for manufacturing, and try to identify a GMP manufacturer as soon as possible so that you have these things integrated in. I'd like to wrap up by saying if none of this made any sense, take a look at our book. We've got a couple chapters on these principles. This is our small company in Michigan, and I'd like to wrap up with the acknowledgement to these folks that have contributed to many of the things I talked about in this short story. Thank you very much.
You know, so in order to keep us on time, I know there will probably be, after that impressive information and history of a nanomedicine that I find compelling. I think, Don, would be okay if we reserve questions for the
That's
question
perfect.
and answer.
Anybody going to brokerage dinner
keep us
tonight?
on
I'll be
time.
there drinking wine with Barrett.
Okay. All right. So
Thank you
so much, Don. We'll have you back on the stage in a minute. So